Good evening. Tonight, 100 things about the deep ocean that nobody has managed to explain yet. Here is the first. In 1997, microphones moored across the Pacific picked up a sound so loud that stations 5,000 kilometers apart heard it at once. Nothing alive is known to make a noise that size. In the summer of 1997, a network of hydrophones anchored across the equatorial Pacific picked up a sound unlike anything in their recordings before. It was brief, deep, and so powerful that sensors nearly 5,000 kilometers apart registered it within minutes of each other. The array had been built to track distant seismic activity and submarine movement, and its microphones normally caught whale calls, ship engines, and the slow groan of the seafloor. This new sound rose quickly in pitch over about a minute, a shape unlike any whale call on record, and far louder than the loudest animal sound ever measured in the ocean. Scientists nicknamed it the bloop, and for years nobody could say what made it. Years later, acousticians at the National Oceanic and Atmospheric Administration compared its shape to recordings of ice cracking apart in the Southern Ocean, and concluded it was probably the sound of a massive iceberg fracturing near Antarctica, an event known as an ice quake. That explanation fit the physics. It did not, however, identify which iceberg, where exactly it broke, or why this single fracture produced a signature so distinctive that it still gets studied on its own, separate from every other icequake ever recorded. In August of 1991, hydrophones listening across the equatorial Pacific began picking up a strange series of sounds nobody had logged before. Short, rising notes repeated over and over in long bouts. Researchers named it upsweep, and it has never really stopped. It still shows up every year, building to its loudest point in spring and again in autumn, a pattern so reliable that acousticians can almost set a calendar by it. By comparing arrival times across widely spaced sensors, scientists traced the sound back to a stretch of seafloor between South America and New Zealand, a remote region dotted with underwater volcanoes and seismic vents. That narrowed things down, but it did not solve the puzzle. Nobody has a research vessel sitting directly above the source when the sound happens, and no camera has ever caught the moment it is made. It could be magma moving through cracks in volcanic rock, or water flashing to steam against superheated stone, or something about the shape of the seafloor itself. 3 decades in, the location is known within a wide radius, and the mechanism is still just an educated guess. On the 1st of March, 1999, every hydrophone in the Equatorial Pacific Monitoring Network picked up the same sound within moments of each other. A smooth, sustained tone lasting about 15 seconds. Analysts working the array that day nicknamed it Julia, after a colleague who first noticed it on the screen. What stood out was not just the sound itself, but how evenly it arrived. Ocean sensors spread across thousands of kilometers of open water usually catch a signal at slightly different times and strengths, letting scientists work backward to a source. Julia showed up almost simultaneously and at comparable volume on every station, a pattern more typical of something enormous moving or fracturing across a wide stretch of ice than a single small point source underwater. The frequency pattern resembled the icequake signals already linked to cracking Antarctic ice, so most researchers place its origin somewhere along that frozen coastline. But no expedition has matched Julia to one specific shelf, glacier, or berg, and the exact mechanics of how a fracture could sound so uniform across such a wide listening area has never been fully worked out. In 1997, the same year that produced several of the strangest recordings in the Equatorial Pacific Archive, hydrophones captured a sound that behaved unlike anything else in the catalog. A low tone that drifted steadily downward in pitch for a full 7 minutes before fading out. Engineers listening to the playback called it slow down, simply because that is what it did. It was not a one-time event. The same descending pattern has resurfaced several times a year since it was first logged. Researchers have compared its shape to sounds already associated with moving ice, the kind of long grinding call produced when a very large mass shifts slowly against rock or against itself. That comparison is reasonable, but it remains a comparison rather than a confirmed match. No one has paired a slow-down recording with a satellite image of ice breaking at the same moment, and no single location has been confidently identified as its source. So the sound keeps a regular place in the yearly data, logged and labeled, while the actual physical event behind that long, patient slide down the frequency scale is still listed as unresolved. Somewhere in the Equatorial Pacific in 1997, one single hydrophone picked up a short, clear, whistling tone that no other station in the network recorded. For almost every other unusual sound in the archive, researchers use a method called triangulation. The same sound reaches several hydrophones at slightly different times, and comparing those tiny delays lets scientists calculate roughly where it came from. The whistle broke that method before it could even start. With only one sensor catching it, there is no second arrival time to compare, no angle to cross-reference, no way to draw intersecting lines on a map and mark a probable source. It is a bit like hearing a single knock through a wall with only one ear pressed against it. You know a sound happened. You cannot say from which direction. Whatever produced the whistle might have been unusually quiet, unusually close to just one station, or positioned in a way that the surrounding seafloor blocked it from reaching any other listening post. No repeat recording has turned up since to give researchers a second chance. It remains the one sound in the collection that is not just unexplained, but structurally impossible to locate with the data on hand. Submarine crews working the Southern Ocean have been describing the same strange sound since at least the 1960s. A low, rhythmic, quacking-like pulse that repeated with almost mechanical regularity, so steady that some sailors assumed it was coming from their own vessel. Whatever it was, it showed up often enough on submarine sonar that crews gave it a nickname, the Quacker, decades before scientists had any real chance to study it properly. For a long time, it stayed a kind of open secret among sonar operators, with no confirmed source. That changed in 2014, when a team of researchers used recording tags attached directly to whales, matched against the timing and location of quacker detections, and linked the sound firmly to the Antarctic minke whale. But identifying the animal answered only part of the question. Nobody has fully explained why minke whales produce this particular repeating pattern, what it communicates, or why it carries such a distinct, almost machine-like rhythm compared to the calls of other baleen whales. The quacking continues through Antarctic waters every year, now correctly credited to its source, its purpose still sitting outside anyone's confident explanation. Since 1989, listening stations across the Pacific have picked up the calls of a single whale, migrating on a normal seasonal schedule, but singing at a pitch that matches no known species. Its calls sit at about 52 hertz, higher than the deep calls of a blue whale and lower than the calls of a fin whale, in a narrow band that does not overlap cleanly with either. Researchers have followed this one voice across decades of recordings, watching it move through the ocean year after year, always alone in the data, never once tracked calling alongside another animal of matching frequency. It picked up the nickname the loneliest whale, and the label stuck. No research vessel has ever gotten a confirmed visual on the animal making these calls, so nobody can say for certain what it looks like, whether a blue whale, a fin whale, or some hybrid of the two. Some scientists think a hearing or vocal difference simply set its pitch apart from birth, though not everyone is convinced that explanation covers the whole pattern. Either way, the calls keep arriving on schedule, still unmatched to any species, still coming from a whale nobody has ever actually seen. In 1997, the same equatorial Pacific hydrophone network that logged several of the ocean's strangest sounds that year picked up a long, low rumble that builds steadily in volume, the way an approaching train seems to grow louder before it comes into view. Engineers labeled the recording the train. Unlike a single sharp event, it stretches out, a sustained wall of low-frequency noise rather than one distinct pulse. Working from the timing of its arrival at different stations, researchers placed its likely source somewhere in the southern hemisphere, in the general direction of the ice shelves ringing Antarctica, the same broad region blamed for several other entries in the same year's recordings. The reasoning follows a pattern that by now feels familiar. Large-scale ice movement can produce exactly this kind of long, building, low-frequency rumble as huge frozen masses grind and shift. But familiar is not the same as proven. Nobody has matched the train to a specific, dated ice event captured by satellite or ship, and the recording has never repeated in a way that lets scientists refine their guess. It sits in the archive as a plausible, unconfirmed story, waiting on evidence nobody has yet collected. Sonar operators working the North Pacific started reporting an odd, twanging sound as far back as the 1950s. A short pulse that dropped and warbled in a way that earned it the plain, practical nickname the bawing among submarine crews who heard it on their headsets night after night. For decades, it stayed mostly a curiosity passed between operators, occasionally speculated about, never formally traced. That changed only in the 2000s, when researchers finally gathered enough recordings paired with visual and acoustic survey data to link the bawing to the minke whale, closing a gap that had sat open for roughly 50 years. Naming the source felt like the end of the story, but it was not. Knowing which whale makes the sound says nothing about why it makes this particular sound, in this particular structure, in these particular waters. Is it a mating call, a spacing signal between males, a way of announcing location across open water? Researchers have proposed each of these ideas at different points, without settling on one. So the bawing sits in an unusual category, a fully identified source, recorded for over half a century, whose actual purpose in the life of the whale remains an open question. In 2014, recorders moored near the Mariana Trench, the deepest known point in any ocean, picked up a sound unlike anything in the existing catalog of whale calls. A call so structurally unusual that the team studying it gave it its own name, the Western Pacific Biot Wang. The sound unfolds in 5 distinct parts, moving from a low moaning tone through a series of pulses before finishing on a bright metallic ring, almost like a struck piece of metal fading out. Researchers matched the overall pattern to Bride's whales, a species known to live in those waters, based on the timing and general shape of the call compared to confirmed recordings. That gave the sound a probable singer. It did not explain the singing. No other whale species produces anything with this 5-part structure or that closing metallic ring, and even among Bride's whales, only some individuals in some areas seem to make it, while others in the same general region never do. Whether it marks a distinct population, a seasonal behavior, or something closer to a regional dialect passed between individual whales is still argued over by researchers, without full agreement. In 1995, the crew of the merchant ship SS Lima sailed for 6 hours through a stretch of the Arabian Sea that glowed white in every direction, from the horizon to the ship's own wake, as if the vessel were crossing a field of snow rather than open water. The light was bright enough on deck to read by. For years, the story sat in maritime records as an odd, hard-to-verify tale, since sailors have reported glowing seas for centuries with no way to check the claim. Then, in 2005, researchers scanning archived satellite images found a matching glow in the same patch of ocean around the same dates. A pale marking on the sensor data covering an area of roughly 15,000 square kilometers, large enough to see clearly from space. An old sailor's story had become a documented event. What still has not been settled is the cause. The leading suspicion involves vast colonies of glowing bacteria coating the surface water, but nobody has sampled one of these events while it was happening, so the exact trigger and why it spread so evenly across such a wide stretch of ocean remains unexplained. Deep below the reach of surface waves, in the ocean layer where sunlight fades into permanent twilight, many small fish spend their days hanging motionless with their bellies pointed toward the surface. Along their undersides run rows of tiny light organs, glowing with a soft downward light tuned to match the faint sunlight filtering down from above. Seen from below by a predator hunting upward, the fish's outline should stand out as a dark shape against a brighter sky. Instead, the glow erases the silhouette almost completely, blending the fish into the light around it. What makes this strange is the precision involved. The brightness of downwelling light changes constantly, with depth, with cloud cover, with the phase of the moon, and with the time of day, yet these fish adjust their own glow to keep pace within moments. Researchers studying hatchetfish and similar species believe the eyes on top of the head somehow measure the light above and feed that reading straight into the light organs below. How that measurement gets translated into exactly the right brightness is a piece of the system nobody has fully worked out. A single bacterium is too small for its own glow to do it any good, and producing light is not free. It draws on the same chemical energy the cell needs for everything else, yet certain marine bacteria keep glowing anyway, sometimes gathering in such numbers that a patch of seawater or a piece of drifting matter shines like a tiny lantern. For a long time, this looked like a pointless expense. The explanation that has gained the most support is almost the opposite of self-defense. Instead of trying to avoid being eaten, these bacteria may be advertising themselves to it. A glowing particle of debris is easier for a small fish or a shrimp to spot and swallow than a dark one, and the gut of that animal turns out to be an excellent place for the bacteria to multiply, warm, sheltered, and full of nutrients. Once the animal moves on and releases waste, the bacteria are carried to a new patch of ocean with a fresh burst of growth behind them. Under this idea, being hunted is not a risk to survive, but a ride to catch. It is a tidy explanation, though still hard to confirm directly in open water. Off the coast of the Pacific Northwest, researchers spent years collecting a small, umbrella-shaped jellyfish called Equoria victoria, cutting the glowing rim from thousands of them by hand to extract a protein responsible for its faint green light. That protein, later named green fluorescent protein, went on to reshape modern biology. Scientists learned how to attach it to other genes, turning it into a glowing tag that lets them watch, in living cells, exactly where and when a chosen gene switches on. Cancer cells, nerve connections, developing embryos, all became easier to track simply by lighting them up green. The work eventually earned a share of a Nobel Prize. And yet, inside the jellyfish itself, the reason for this protein's existence is still not settled. The animal's light organs first produce blue light through a separate chemical reaction, and the green protein absorbs that blue light and re-emits it as green before it ever leaves the body. Why the jellyfish would go through the trouble of shifting its own light from blue to green, what difference that color makes to it, has never been convincingly explained, even by scientists who built careers on borrowing the protein for other purposes. Off the coast of New Zealand, along an undersea ridge called the Chatham Rise, researchers hauled up 3 species of deep-sea shark in 2021 and found, almost by accident, that all 3 produced their own light. One of them, the kitefin shark, reaches almost 2 meters in length, making it the largest animal with a backbone yet confirmed to glow on its own. Its entire lower body emits a faint blue light, likely for the same reason smaller fish use it, to erase its outline from anything hunting below. What surprised the team was not the glow itself, but how long it had gone unnoticed in an animal that size, hauled up by fishing boats for years without anyone testing it in full darkness. Sharks, built of cartilage rather than bone and living far deeper than most study sites, are simply harder to bring up alive and examine properly. The chemistry behind the shark's glow also appears to run through a different pathway than the one used by glowing fish and invertebrates, tied to hormones already known to affect its skin. Exactly how that system evolved and how many other deep-sea sharks share it is still being worked out. During daylight hours, certain patches of coastal water turn a dull reddish pink, thick with a single-celled organism called Noctiluca, sometimes nicknamed sea sparkle. The color comes from a pigment packed inside each tiny cell. Wait until after dark, disturb that same water with an oar, a hull, or a breaking wave, and it answers with flashes of electric blue, thousands of individual cells lighting up at once wherever the water is stirred. The same organism, the same patch of ocean, 2 completely different displays depending on whether the sun is up. Sailors and coastal residents in places like the Arabian Sea have described both versions for generations. In recent decades, these blooms have been showing up more often and covering more ground than older records suggest, spreading into waters where they were once rare. Scientists tracking the change have looked at shifting water temperatures, changing oxygen levels, and nutrients washing in from land, without settling on a single clear cause. Noctiluca does not need sunlight to feed the way ordinary algae do, which already sets it apart. And why its blooms are expanding now, in these particular places, remains a question researchers are still chasing rather than one they have answered. Certain small shrimp living in the middle depths of the ocean have an unusual response to being grabbed. Rather than fighting back or swimming away, they release a burst of glowing chemical fluid into the water, leaving behind a bright blue cloud roughly the shrimp's own size, while the animal darts off into the dark. The obvious explanation, that the cloud simply confuses or startles the attacker, only covers part of the picture. Researchers studying the behavior have proposed something more layered, that the burst of light is aimed less at the immediate attacker and more at whatever else happens to be nearby. A sudden bright cloud in open water draws attention from a distance, and the idea is that it may attract a larger predator toward the disturbance, one big enough to see the first attacker as a meal in its own right. In the confusion, the shrimp slips away unnoticed. It is a clever piece of reasoning, built from lab observation and the design of the light-producing organs involved. But confirming it in the wild would mean witnessing this exact three-way encounter unfold naturally at depth, something almost no one has managed to do. Trawl through the open ocean at almost any depth below the sunlit surface layer, and roughly 3/4 of the animals brought up in the net, fish, shrimp, jellyfish, worms, will turn out to produce their own light. Across the deep sea as a whole, glowing is closer to the rule than the exception. Now compare that to a lake or a river. Freshwater bioluminescence barely exists. A handful of odd exceptions turn up here and there, a certain snail in New Zealand streams, some fungus gnat larvae in caves, but there is no freshwater equivalent of the glowing fish, squid, and jellyfish that fill the ocean's middle depths. The two environments are both dark once you get away from the surface. Both hold plenty of predators and prey, and yet only one of them is full of living light. Researchers have floated a few possible reasons. Differences in water chemistry, the fact that lakes and rivers are geologically much younger than ocean basins, or simply that freshwater animals evolved along a different path and never picked up the trick. None of these explanations has been shown to be the real one, and the sharp divide between salt water and fresh water remains unresolved. Most creatures that glow in the deep sea, and most eyes that watch for that glow, work in shades of blue and green, the only colors that travel far through seawater. Red light fades out almost immediately, which is exactly why a small, sharp-toothed fish called the black dragonfish uses it. Below each eye sits a small organ that shines with a deep red beam, closer to the color of a dying ember than anything else glowing nearby. To almost every other animal at that depth, the beam is invisible, since their eyes cannot register red at all. The dragonfish can flood the water ahead of it with light, hunt by that light, and stay unseen by prey and predators alike, a private channel no one else is tuned to. What makes the fish itself unusual is that it can see its own red beam, using a visual pigment built from a borrowed, chlorophyll-related compound that most likely comes from its diet rather than from its own chemistry alone. Vision built this way is rare enough that researchers treat it as one of the strangest solutions in the deep sea, and exactly how the fish first assembled this trick remains only partly understood. When a deep-sea jellyfish called Atala finds itself caught in the grip of a predator, it does not try to swim away. Instead, a ring of blue light begins to spin around the rim of its bell, one flash chasing the next in a rapid pattern that looks less like a warning flare and more like a tiny pinwheel spinning in the dark water. The display seems built to draw attention from a distance, and the leading idea among researchers is that it works as a call for help, summoning something larger toward the jellyfish's attacker, in the hope the bigger animal treats the attacker as the more interesting meal. The pattern proved so effective that scientists built an artificial copy, a small spinning ring of blue lights mounted on deep-sea camera rigs, and used it to draw curious animals close enough to film, including rare footage of a giant squid in its own habitat. What the imitation has never explained is the shape of the real thing. A steady glow or a simple pulse might seem to serve the same purpose, and why evolution settled on a spinning ring specifically rather than any simpler pattern is still an open question. During the Second World War, Navy sonar crews kept running into a seafloor that had no right to be there. Their echo sounders bounced off something solid roughly 400 meters down, in open water where charts showed the real bottom lying far deeper. Stranger still, this false floor refused to sit still. It rose through the evening, sometimes climbing to within a few dozen meters of the surface, then sank back into darkness before dawn. For years, crews debated what they were seeing, blaming faulty gear or some unmapped ridge that somehow shifted with the tide. By 1948, scientists at the Scripps Institution of Oceanography solved it by lowering nets straight through the layer. Not rock or mud, but a living haze of fish and drifting crustaceans, spread across entire ocean basins and thick enough to throw back sound. They named it the deep scattering layer. Even now, an echo bouncing off that layer cannot tell a researcher exactly what animals are inside it. Sound alone cannot tell a swarm of shrimp from a shoal of small fish, so what a given sonar trace is detecting still has to be confirmed the old-fashioned way, by hauling something up and looking at it. Every evening, a mass of ocean animals estimated in the billions of tons leaves the deep water and swims toward the surface, then reverses course before sunrise and swims back down. It happens in the Pacific, the Atlantic, the Indian Ocean, everywhere between, all on the same daily rhythm, making it the largest movement of life on the planet, larger than any migration of birds or land mammals. Scientists call it the vertical migration. Unlike a bird migrating between continents once a year, this journey happens twice a day, across nearly the entire open ocean at once. Small crustaceans, squid, and lanternfish rise hundreds of meters toward the surface to feed under cover of darkness, then retreat to darker water before light returns, to avoid animals that hunt by sight. What keeps the figure hard to pin down is scale. Newer surveys using more sensitive sonar keep revising the biomass sharply upward, suggesting the true number may be several times larger than textbooks claimed a decade earlier. No one now defends a single number as final. The ocean is too large, too deep, and too dark for any survey to count every participant, so the true nightly tonnage remains an estimate, not a measurement. As afternoon fades into evening across the open ocean, the trigger for the largest migration on the planet is not a clock or a current, but simply the dimming of light. Animals scattered through the twilight zone track the fading sun with startling precision, holding themselves at a specific brightness level as it sinks deeper, riding it upward like swimmers following a fixed depth of shade. Researchers call this a preferred light intensity, and animals seem to defend it more tightly than instruments of past decades could measure. A cloud passing over the surface far above, or a heavy overcast sky, can send the whole layer rising early, long before actual sunset. Bright moonlight on a clear night can push it back down. What nobody has fully worked out is the mechanism behind that precision. These are small animals, many without anything resembling a complex eye, yet as a group they track light changes of a fraction of a percent well enough to hold a shared depth across a shifting crowd of countless individuals. Whether each animal senses the threshold on its own or responds to the movement of its neighbors, and how that signal could travel fast enough through open water, remains unresolved. Small, silvery fish covered in rows of light-producing organs may be the strangest candidate for the most numerous vertebrate on Earth. Lanternfish, named for the pattern of glowing spots along their sides, live by the trillion through the twilight zone of every ocean, rising each night to feed and sinking again by morning as part of the great migrating layer. They rarely grow longer than a hand's width, yet their numbers make them a central food source for tuna, squid, seabirds, and whales across the planet. For decades, textbooks listed the world's lanternfish weight at a modest figure, based on old trawl surveys. Then a research voyage that circled the globe using sonar instead of nets returned an estimate roughly 10 times higher, arguing that fish this small and fast dodge conventional nets more easily than anyone assumed. That study reshaped thinking about how much life the open ocean can support, and how much carbon and energy moves through these fish daily. Yet the two estimates have never been reconciled. Depending on which survey a scientist trusts, lanternfish either outweigh nearly every other vertebrate group on the planet combined, or they are merely a large piece of a still uncertain total. Every night, as the deep scattering layer rises to feed near the surface and sinks again before sunrise, it carries something besides itself down into deep water: carbon. Animals that feed near the surface digest and breathe out carbon dioxide at depth, release waste that sinks, and occasionally die and drift downward, moving carbon from shallow water, where it could easily return to the atmosphere, into the deep ocean, where it can stay locked away for centuries. Scientists call this the biological pump, and the portion driven specifically by migrating animals is sometimes called the migrant pump. Early calculations suggested it moved a meaningful slice of the ocean's carbon budget downward every single day, across every ocean basin at once. The trouble is measuring it precisely. Following billions of small, fast-swimming animals through a full day and night cycle, across water kilometers deep, is far harder than sampling a slow drift of dead material falling from above. Some research teams estimate the migrant pump accounts for only a small slice of deep carbon transport, while others argue it may rival or exceed the sinking of dead material entirely. That gap between estimates remains one of the largest open questions in the entire ocean carbon budget. Pull a fine net through open water almost anywhere on the planet, at almost any depth below the sunlit zone, and there is a strong chance the most common animal caught will belong to a single genus of small, dark, torpedo-shaped fish called cyclothone. Some researchers consider them the most numerous vertebrates on the entire planet, outnumbering every bird, every shallow water fish, every mammal by a wide margin. Yet almost nobody has ever seen one alive in its natural state. Cyclothone live at depths where sunlight never reaches, and the fine mesh nets used to catch them for study crush their soft, delicate bodies on the long haul to the surface. So scientists studying them mostly examine bruised, lifeless specimens rather than living fish. A handful of glimpses from deep-diving submersibles and remotely operated cameras have caught them drifting in place, but such sightings remain rare, brief, and limited to the few locations where research vehicles happen to be looking at the right moment. Basic questions that would be simple to answer for almost any land animal, such as how they behave, hunt, or interact with each other, are still largely guesswork for what may be the single most abundant vertebrate lineage alive today. Not every animal in the deep scattering layer joins the nightly rise toward the surface. Careful sampling at different depths, across many species of small crustaceans, squid, and lanternfish, keeps turning up individuals that stay behind in deep, dark water while most of their population swims upward to feed. These animals are not sick or dying. Many appear healthy, simply resting at depth through an entire cycle of darkness and daylight, while their neighbors make the long swim up and back. In some populations, only a small fraction skips the journey. In others, resident individuals make up a much larger share. Researchers have proposed several explanations. Perhaps these are animals that fed recently and have less reason to make a risky trip toward the surface, where more hunters can see them. Perhaps age, size, or reproductive state changes the calculation of risk versus reward for a given individual. Perhaps some are too weak to complete the swim. None of these explanations has been confirmed as the main driver, and different studies of different species sometimes point in different directions. Why a portion of an otherwise identical population opts out of the single largest migration on the planet, night after night, is an open question. North of the Arctic Circle, for weeks in deep winter, the sun never rises at all, yet the deep scattering layer keeps moving on schedule. Researchers once assumed the migration would simply stop once daylight disappeared, since the behavior was thought to depend on tracking the sun. Then teams working in the fjords of the Norwegian archipelago of Svalbard began leaving sonar instruments moored through the dark season, and the recordings told a different story. Zooplankton and small fish kept rising and sinking on a roughly daily rhythm through weeks of total solar darkness, timed instead to the fainter light of the moon. As the moon moved through its phases, the migration pattern shifted with it, sinking deeper during the brightest full moons as if avoiding being seen, then rising again as the moon waned. During especially bright moonlit stretches, some animals reduced their rise nearly entirely, staying low as though moonlight alone posed enough risk. That such a faint light source could reorganize the behavior of an entire ecosystem through total polar darkness surprised the researchers who found it. Exactly how sensitive these animals are to moonlight and how they tell it apart from other faint sources is still being worked out. Beneath a solid ceiling of Arctic sea ice, thick enough to walk across, almost no sunlight reaches the water below, especially through the winter months. Yet instruments moored under that ice for months have recorded the deep scattering layer still rising and sinking close to its usual daily schedule, even when surface light readings sit at nearly nothing. That finding puzzled researchers, since the migration is generally explained as a response to changing light. Under thick ice and snow cover, the light penetrating the water can be too faint for ordinary instruments to register at all, let alone faint enough to explain animals adjusting their depth by dozens of meters on schedule. A few explanations have been proposed. Perhaps some animals retain an internal sense of daily rhythm that keeps running without a clear external cue, similar to a body clock. Perhaps their eyes, adapted for the near-total darkness of deep water, can detect traces of light too faint for any instrument lowered from a ship to pick up. Neither explanation has been confirmed. What keeps a layer of animals moving on schedule under a frozen, lightless ceiling for months on end remains one of the more puzzling parts of the migration. Inside the deep scattering layer, countless small animals are hunting and being hunted, yet the details of who eats whom through the middle of that food web remain surprisingly sketchy. Part of the problem shows up the moment a researcher cuts open a freshly caught specimen, expecting to find a clue. More often than not, the stomach is empty. Many of the fish and crustaceans that make the nightly journey digest their meals so quickly, sometimes within a couple of hours, that by the time a net brings them to the surface, any trace of what they ate is already gone. Some species feed almost exclusively at night near the surface, which only narrows the sampling opportunity further, since a trawl has to catch them at exactly the right moment in an already narrow window. Chemical tracers drawn from body tissue can hint at a general diet over weeks, but cannot say what a given animal ate on a given night. As a result, entire layers of the food web connecting the smallest drifting animals up through lanternfish, squid, and the predators that follow them remain mapped only in rough outline, with plenty of links still guessed at rather than confirmed by direct observation. For most of the 1800s and the century that followed, the giant squid was a creature known only in pieces. Fishermen found tentacles tangled in their lines, carcasses washed on beaches, and half-digested bodies pulled from the stomachs of sperm whales. Scientists could measure it, dissect it, name it Architeuthis, and still had never watched it move through open water. That changed only in 2012, when a Japanese and American team spent weeks lowering cameras into deep water off the Ogasawara Islands, south of Japan. A lure blinked in the dark at roughly 900 meters down, and eventually a giant squid drifted into view, silver and calm, its long arms coiling slowly around the bait before it swam back into the dark. It was the first time a giant squid had been filmed alive in the place where it actually lives. And that single encounter is nearly everything anyone has to go on. How the species hunts, how it finds a mate in total darkness, how long an individual actually lives. A century and a half of dead specimens taught us its shape. One evening of footage barely began to teach us its life. In March of 2025, a small robotic submarine drifted through cold water near the South Sandwich Islands, and its camera caught something researchers had never recorded in the wild before. A live colossal squid, drifting at around 600 meters down, its body almost glassy and clear. The animal was young, no longer than a school ruler, but its species is thought to be the heaviest invertebrate alive, with mature adults estimated to reach weights far beyond any giant squid. Unlike its more famous cousin, the colossal squid carries hooks that rotate on swivels along the middle of its tentacles, hardware no other squid on Earth possesses. For decades, this animal was known from carcasses hauled up in fishing nets or pulled, half digested from the stomachs of sperm whales, which hunted often in deep water. A fully grown adult has still never been filmed in its own habitat. Scientists cannot say how it hunts, how it finds a mate in water with almost no light, or how long it takes to grow from that small glassy juvenile into an animal that can weigh as much as a small car. Almost the entire life of the colossal squid remains a closed door. Near the Gulf of Mexico in 1998, a submersible camera recorded a squid unlike anything in the reference books. Long, thin arms that did not taper smoothly like a normal squid's, but bent sharply partway down, held out at right angles like a set of elbows. Researchers named the genus Magnapinna, the bigfin squid, and kept watching. Since that first sighting, industry and research cameras have recorded it maybe a couple of dozen times in total, drifting at depths from a few hundred meters to several thousand, its elbowed arms trailing beneath it like loose threads. Every one of those sightings is video or a photograph. Not one adult bigfin squid has ever been caught or brought to the surface for a scientist to examine. The only physical specimens in hand are a handful of damaged juveniles, too young and too different in shape to explain what the elbows are even for. Are they used to walk along the seafloor, to sense vibrations, to snare prey drifting past in the current? Nobody studying Magnapinna can say, because nobody has ever watched an adult one closely enough to find out. It remains a full-grown mystery that science has only ever seen from a distance. Deep-sea cameras drift past something that barely looks like an animal at all. A huge, loose, transparent sheet folding gently through the water, more like a plastic bag caught in a current than anything with a nervous system. This is Deepsteria, a genus of jellyfish named after the research submersible that brought one into view decades ago. It has almost none of the features people expect from a jellyfish. Instead of a dome with trailing tentacles, its body is a thin, wide bell that can billow open and then fold closed around whatever drifts inside it. Sightings are rare enough that each new piece of footage travels widely among researchers. Because Deepsteria is almost never seen in the act of feeding, and because it has no obvious eyes, no head, and only a simple nerve net instead of a brain, scientists cannot explain how it senses that something edible has drifted close enough to trap. Does it react to touch alone, to chemical traces in the water, or to some form of pressure change? Even after repeated encounters on camera, the animal that looks the least like a hunter may be one of the most effective, and nobody understands how it manages the trick. Researchers lowered a camera onto the carcass of a gray whale resting on the seafloor in Monterey Canyon in 2002, expecting to document the usual scavengers. Instead, they found the bones themselves covered in something new to science. Small red worms with no mouth and no gut, feeding directly on the skeleton by dissolving it. Named Ostax, Latin for bone eater, the worms grow root-like structures that burrow into whale bone, and with help from bacteria living inside their tissue, break down the fats and proteins locked within. The stranger discovery came next. Every visible worm turned out to be female, and inside the gelatinous tube surrounding each one, scientists found dozens of males living there, microscopic, never developing past a larval stage, there purely to fertilize her. A single female can carry a small colony of these dwarf males for her entire life. Ozdax was found entirely by accident, on a whale fall nobody had planned to study so closely, which raises an obvious question. How many other whale skeletons scattered across the ocean floor are hosting the same hidden colonies, unseen, simply because no camera has drifted past them yet? Nobody studying the deep sea floor can honestly answer that. For roughly a century, whenever a trawl net brought a barrel-eye fish up from deep water, scientists found the same disappointing thing: a crushed, ruined head where a transparent dome should have been. The pressure change and the net destroyed the one feature that made the fish worth studying before anyone could see it intact. Only in 2009 did researchers studying footage from a deep-diving robotic camera finally get a clear look at a living barrel-eye, drifting undisturbed a few hundred meters down with its fluid-filled, transparent dome intact. Inside that dome sat two tubular, barrel-shaped eyes, and the footage revealed something no crushed specimen could ever have shown. The eyes could rotate freely inside the transparent shield, swiveling upward to watch for prey silhouetted above, then swinging forward when the fish turned to feed. A face, essentially, hidden behind its own window. The discovery solved one old puzzle and opened another. Researchers still debate how the barrel-eye's dim, narrow vision translates into successful hunting in near-total darkness, and how a fish with such a fragile, fluid-filled head survives collisions and pressure changes. A century of broken specimens had hidden a working eye that nobody had ever properly seen move. The pelican eel looks like a mistake of proportions. A thin, dark, ribbon-like body trailing behind a mouth so large it makes up a huge share of the entire animal, hinged loosely enough to swing open far wider than its head. When fully open, that mouth looks capable of swallowing prey several times the eel's size, which is how the species earned its other common name, the gulper eel. Yet when researchers have examined the stomach contents of captured specimens, they find small crustaceans and tiny fish, nothing close to the giant meals the anatomy seems built for. That mismatch between the tool and its apparent use is still not resolved. Some scientists suspect the huge mouth works like a net, expanding to trap swarms of small prey in a rush of water rather than swallowing large animals whole. Others think it may be built for rare opportunities, large meals that are almost never observed because they happen so infrequently. Either way, nobody has explained how the thin tissue of that mouth avoids tearing when it stretches so far past its resting size, or why an animal that mostly eats small prey evolved a mouth so out of proportion to its diet. After more than 2 decades of footage sitting in research archives, marine biologists gave a name in 2024 to an animal that had been puzzling them since the year 2000. A soft, transparent, swimming sea slug that behaved like nothing else in its family. Bathydevius caudactylus drifts through the deep using a wide, gelatinous hood, and when prey drifts inside that hood, the animal folds it closed, much the way a land plant closes around an insect. It can produce its own light, glowing faintly when disturbed, a trait almost never combined with a swimming, hooded body plan. When researchers compared its anatomy against every known group of sea slugs, it did not fit cleanly into any of them. It was different enough that scientists had to create a new family to hold it, a rare step in a field where most new finds slot into categories that exist already. Video of the animal existed for years before anyone worked out what they were looking at. Its closest relatives, how widespread it is across the world's oceans, and why this lineage evolved a hunting method found almost nowhere else among mollusks, are still open questions, unanswered even after its formal description. Long before sharks looked anything like they do now, one branch of their family tree broke away and went its own direction, roughly 400 million years ago. That branch became the chimeras, sometimes called ghost sharks, cartilaginous fish with smooth skin, huge dark eyes, and a single feathery fin down the spine, built for a slow glide through cold, deep water rather than a fast chase. Because they live far deeper than most sharks and tend to avoid nets and bright lights, chimeras were known to science almost entirely through dead specimens. It was only in 2009 that researchers reviewing years of deepwater video had enough footage to confirm and formally describe a living chimera moving through the canyon it called home, undisturbed, feeding quietly along the bottom in near total darkness. The name ghost fits in more than one way. Chimera flesh, skin, and cartilage rarely preserve well, so even the fossil record of their ancient relatives is thinner than it is for sharks, leaving long gaps in their story. Basic details of how most living chimera species reproduce, how long individuals live, and how many separate species exist in deep water around the world remain unresolved. Behind the public galleries of most natural history museums sits a much less visited room. Rows of shelving lined with glass jars, each one holding an animal pulled from deep water during a research expedition, preserved in fluid, and labeled with only a date, a location, and a depth. Some of those jars have sat untouched since expeditions that happened decades ago. The animal inside might have a name, or it might be new to science, indistinguishable from a known species until someone with the expertise sits down, compares it against everything described, and writes up the differences in a formal paper. That work is slow, and there are far more jars than there are specialists trained to open them. Marine biologists who study the deep admit they cannot say how many of the species sitting in storage worldwide are still waiting for a name. Estimates for how much of ocean life remains undiscovered vary between research groups, and nobody treats any single number as settled fact. Which means some of the strangest, least understood animals in this chapter may not be hiding in the deep ocean. They might already be sitting in a jar, a few meters from a hallway, waiting their turn. On the floor of the Gulf of Mexico, beneath roughly a kilometer of ocean, sits a lake that should not exist. Submersible pilots who found it nicknamed it the Jacuzzi of Despair, because it looks like a spa pool resting on the seabed, with its own shoreline, its own surface, and slow ripples drifting across it. The water inside is brine, several times saltier than the seawater around it, squeezed out of salt deposits left behind when this stretch of ocean floor was once a shallow sea drying under the sun. That brine is so much denser than ordinary seawater that it refuses to rise and blend, sitting instead inside a far larger body of water, its edge sharp enough to trace with a camera light. Mussels and tube worms crowd the rim, feeding on chemicals seeping through the mud, tolerant of conditions that would kill most life. Dozens of these pools are now known across the Gulf floor, and new ones keep turning up as vessels map terrain nobody has examined closely. Why the boundary stays so crisp instead of slowly smearing into the water above, the way mixing physics would predict, is still something nobody has fully explained. Submersible pilots working the brine pools of the Gulf of Mexico learn to slow down well before they get close. These pools of salty water sit in shallow basins, dense enough to hold a shoreline and surface of their own, and a vehicle approaching too fast pushes a wave ahead of it that breaks against the far shore, the same way a wake rolls onto a beach. What waits along that shore is stranger still. Crabs, fish, and shrimp wander into the brine, drawn perhaps by microbial mats growing along the edge, and the water stops them almost instantly, several times saltier than their bodies can tolerate. Their bodies stay intact rather than decaying, preserved by the same chemistry that killed them, collecting into what researchers call a ring of death, a border of still animals marking exactly where ordinary ocean ends. Cameras have followed the ring around entire pools without a gap. What is strange is how rarely any animal makes it back out, even though open water sits only a body length behind. Whether that comes down to shock, the density of the fluid, or some chemical effect nobody has isolated, remains unresolved. In 1965, a research ship called the Meteor lowered instruments into a depression on the Red Sea floor and pulled up readings that made no sense. Water that deep should have been near freezing, like almost everywhere else on the ocean floor. Instead, the instruments found pools of hot brine sitting more than 2,000 meters down, in basins later named Atlantis Deep and Discovery Deep. Readings taken in the hottest basin over the following decades recorded temperatures above 60 degrees Celsius, warm enough to scald skin, and the heat has kept climbing as it bleeds up from the crust below. The brine carries no oxygen at all and is thick with dissolved metal, enough zinc, copper, and silver that mining companies have surveyed the mud beneath it for the ore. By every ordinary rule, that combination should leave the water sterile. It has not. Microbes pulled from the hottest layers appear to run their chemistry on the metals themselves instead of avoiding them, something closer to breathing metal than breathing air. How they keep their proteins from falling apart in that heat is still being sorted out one sediment core at a time. In 1977, a submersible called Alvin descended toward the Galapagos Rift, a crack in the seafloor northwest of South America, carrying scientists expecting nothing but bare rock and cold water. Instead, their lights picked out towers of dark mineral rising off the seabed, venting water that shimmered like smoke. Vents like these have since been measured pushing out water well over 300 degrees Celsius, hot enough to melt lead, yet it never boils because the weight of the ocean above keeps it liquid. Around the vents, the crew found thickets of life with no business existing in total darkness. Tube worms taller than a person, beds of clams, crabs picking through the debris, all running on chemical energy from the vent water instead of sunlight. Until that dive, most scientists assumed every food chain needed sunlight somewhere near the bottom. The Galapagos vents broke that assumption in a single expedition. What is still not settled is how many separate times life has independently taken root around vents like these, and whether chemistry like this could explain how life on Earth got started in the first place. In the year 2000, a research vessel mapping the middle of the Atlantic Ocean found a field of towers on an underwater mountain called the Atlantis Massif, far from any volcano. Scientists named the site the Lost City, and the name fits. Its tallest spire reaches roughly the height of a 20-story building, built from pale carbonate rock, not the dark minerals of most vent fields. What makes it strange is not just its size, but its fuel source. Most hydrothermal vents run on heat from magma, yet this one sits far from any active magma chamber. Its heat instead comes from a slow reaction called serpentinization, seawater soaking into mantle rock and reacting with it, releasing heat and hydrogen gas with no volcanic activity. Chemical dating suggests parts of the system have run continuously for more than 100,000 years, building rock without ever erupting. Microbial mats coat the towers, apparently living off hydrogen alone, with no sunlight and no magma feeding them. Because the same reaction could happen on icy moons with buried oceans, scientists studying it are also asking whether something similar runs right now under the ice of Europa, unseen and unproven. Divers exploring flooded caves on Mexico's Yucatan Peninsula sometimes swim through perfectly clear fresh water and hit what looks like a wall of frosted glass hanging in the cave. It is not glass and it is not solid. It is a halocline, the boundary where fresh water on top meets salt water underneath, resisting mixing because of the difference in density between the layers. Light bends oddly crossing that boundary, blurring anything on the far side the way heat shimmer blurs a road in summer, which is why divers describe swimming through glass or sometimes oil. A similar boundary forms a visible skin over brine pools on ocean floors worldwide, separating ordinary seawater above from dense salt water below, sharp enough that a submersible's arm appears to blur crossing through it on camera. Reach a hand through a halocline, and the water on the other side feels heavier and warmer within a few centimeters. What keeps that boundary so thin and sharp, rather than smearing into a gentle gradient the way diffusion would predict, is still not fully explained, and different caves hold their glassy layers intact for very different lengths of time before finally fading. Lower a sensor into the Black Sea past roughly 150 meters, and the oxygen readings drop to nothing and stay there all the way to mud more than 2 kilometers below. That makes the Black Sea the largest oxygen-free body of water on the planet, mostly dead water sitting beneath a thin living layer near the surface. The reason traces back to how it connects to the world outside it. Only a narrow, shallow channel through the Bosphorus links it to the Mediterranean, letting in a thin ribbon of salty water that stays near the top, while heavier river water from streams like the Danube settles underneath, and the two barely mix. That stratification has held for thousands of years, since Mediterranean water first breached in and drowned what had been a freshwater lake. Wooden shipwrecks over 1,000 years old have turned up in that dead zone almost perfectly intact, masts still standing, since nothing down there decays wood the way oxygen would elsewhere. What keeps the layers from ever fully turning over, the way lakes do elsewhere, is understood only in outline, and researchers argue over how far the oxygen-free layer has crept toward the surface in recent decades. Pull a chunk of sediment from certain deepwater zones, hold a flame to it, and the ice inside catches and burns while the rest of the block stays cold and wet. That flammable ice is a gas hydrate, a cage of water molecules trapping methane inside it, stable only under the cold and crushing pressure of deep ocean sediment or permafrost. It looks and feels like ordinary ice until somebody lights it. Long stretches of the world's continental margins hide layers of it under the mud, and drilling surveys keep finding more in places nobody expected. The trouble is nobody agrees on how much is actually down there. Estimates of the total methane locked in seafloor hydrates worldwide have varied by several times over between research teams, depending on drilling data, sonar surveys, and models of gas movement, and the range has never narrowed. The gap matters because methane trapped in hydrates is a greenhouse gas far more potent than carbon dioxide, and warming bottom water could, in theory, destabilize the shallowest deposits. Whether a release like that has happened before in Earth's past, and how easily it could happen again, remains an open question among the scientists who study the seafloor. In 2010, researchers surveying the Black Sea floor near the Bosphorus Strait found something that looked, on their instruments, like an ordinary river, except it ran along the seabed under nearly 100 meters of water. It had banks on either side, a winding channel cut into the mud, even small rapids where the flow sped up over a drop in the seabed. What moves through the channel is not fresh water, but a dense current of cold, salty, sediment-heavy water sliding downhill into the deep basin, hugging the bottom because it is heavier than the water around it. Measured for volume, researchers calculated that if this river ran across open land instead of the seafloor, it would rank as the 6th largest river on Earth by flow. Similar channels have since turned up in the floors of other seas and in canyons off major river deltas, carrying sediment into deep water in fast pulses rather than a steady drift. How much of the ocean's sediment, nutrients, and pollution travels this way, hidden inside currents nobody can see from the surface, is still being mapped, and researchers suspect many more such channels remain undiscovered. When researchers lowered instruments into a pool called cryos in the eastern Mediterranean more than 3 kilometers down, they found water close to pure magnesium chloride brine, a chemistry so aggressive most assumed nothing could be alive in it. Magnesium chloride at that concentration does not just make water salty, it pulls proteins apart at the molecular level, unraveling the machinery any ordinary cell needs to function. Early sediment traps meant to catch material drifting into cryos came back essentially empty, reinforcing the idea the basin was sterile. Then researchers analyzing samples more carefully found genetic traces of microbes inside the brine, not simply washed in dead from above, but present in a form consistent with life. Nearby pools named Discovery and Urania turned out to hold similar traces, each basin with its own chemistry and its own residents suited to it. Whether the microbes in cryos are truly alive, slowly ticking over in the dark, or whether what was found is a chemical trace of cells that died the moment they drifted in, is still argued over by scientists who study these basins. Either answer would push the known limit of what salt concentration life can tolerate further than most once thought possible. In 2018, an underwater robot crossed the floor of the Black Sea and found a merchant ship from ancient Greece lying on its side whole. The mast was still standing. Both steering oars were still in place at the stern. Archaeologists dated the vessel to roughly 2,500 years old, the oldest known intact shipwreck on Earth at the time. The reason is chemistry, not luck. Below a certain depth, the Black Sea holds almost no oxygen, because fresh river water sits on top of heavier salt water and the two layers barely mix. Without oxygen, the bacteria that normally rot cannot live, and neither can the shipworms that hollow out hulls in other seas. So the vessel sat there in the dark and the cold, essentially unchanged, while more than 2,000 years passed on the surface above it. The ship's shape matches a type of vessel painted on Greek pottery, but until this find, no one had ever seen one in 3 dimensions. Researchers still cannot say how common this design once was, how the ship ended up on the seabed, or how many more like it are resting in that same lightless water. Divers working in the Baltic Sea regularly find wooden warships and trading vessels from the 1500s still standing upright on the seabed, with carved decoration on the hull and rigging still coiled on deck. Some look almost ready to sail. The reason has nothing to do with cold alone. In the Atlantic and the North Sea, a wood-boring mollusk called the shipworm can reduce a sunken oak hull to a scatter of splinters within a few decades. The Baltic is different because so much fresh water flows in from surrounding rivers that the sea stays only lightly salted, and shipworms cannot tolerate that low salinity. Without them, wood that would vanish elsewhere simply stays wood, sometimes for over 400 years. Survey teams using sonar have mapped dozens of these vessels across the Baltic floor, many of them merchant ships with no flag, no cargo manifest, and no name anyone can match to a record. Researchers can measure the timbers, count the gun ports, and study the carving in fine detail, yet still have no idea who built the ship, what it carried, or where it was headed when it went down. On a clear morning in July of 1909, the steamship Warata left Durban on the coast of South Africa, bound for Cape Town with 211 passengers and crew aboard. She was never seen again. Another ship spotted her smoke on the horizon that evening, and then nothing. No wreckage washed ashore, no lifeboat was found, no body was ever recovered, despite the route running close to a coastline with active ports on either end. The Warata was a large, modern steamer for her time, and questions about her design had already been raised before that voyage, including concerns that she rode top-heavy and recovered slowly when she rolled in rough seas. A storm did pass through the area around the time she disappeared, but a storm alone does not usually erase a ship of that size without leaving a single piece of debris. Search expeditions have combed the seafloor off the South African coast for more than a century, chasing wreck sites that turned out to belong to other vessels entirely. The actual resting place of the Warata has never been confirmed. Her passengers' families never received so much as a splinter to bury. In March of 1918, the USS Cyclops left the island of Barbados carrying a full load of manganese ore, more than 300 people, and a course set for Baltimore. She was a naval collier, built to haul coal and ore out to warships at sea, and by every account she was running normally. She never reached port, never sent a distress call, and no wreckage from her has ever been positively identified, which makes her disappearance the largest loss of life in United States Navy history outside of direct combat. Investigators considered several explanations. The manganese ore she carried was heavy and could shift if loaded badly, which might have made the ship unstable. A storm crossed her likely path. Some officers even wondered about wartime sabotage, since the country was at war and one of her officers had a disputed background. None of these explanations were ever confirmed because there was nothing left to examine. The ship's route lay within busy, well-charted shipping lanes, not some remote corner of open ocean, which is part of why the case still unsettles naval historians. A steel ship longer than a city block does not usually vanish without a trace. In 1901, sponge divers working off the small Greek island of Antikythera found a shipwreck scattered across the seabed, full of bronze statues and other cargo bound most likely for a wealthy buyer in Rome. Among the corroded lumps they brought up was a fist-sized block of bronze that sat in a museum drawer for decades before anyone noticed it was full of gears. Later study, including modern scanning, revealed dozens of interlocking bronze gears inside, cut with a precision that would not become common again for well over 1,000 years. The device tracked the movements of the sun and moon through the sky, predicted eclipses, and even accounted for the irregular timing of the ancient Olympic Games. Researchers now call it the world's first known analog computer. And yet the basic questions remain unanswered. No signature, no maker's name, and no instructions survived with it. Historians have proposed workshops on Rhodes or a connection to the astronomer Hipparchus, but nothing is proven. No second example of this device, or anything close to it in complexity, has ever turned up anywhere else from the ancient world. In March of 2022, a search team in the Weddell Sea off Antarctica lowered cameras through more than 3 kilometers of freezing water and found the wooden ship Endurance sitting upright on the seabed, looking almost the way she did the day she sank. Ernest Shackleton's ship had been crushed by pack ice and went down in that same sea back in 1915, and the wreck was found remarkably close to the position his navigator had calculated using only a compass, a chronometer, and the stars. The timbers were intact. The ship's wheel still stood at the stern, and the name Endurance was still readable across the wood. Part of the preservation comes from the extreme cold, and part of it comes from the near total absence of wood-eating organisms at that depth in that stretch of ocean. What researchers have not fully settled is why those organisms are so scarce there specifically, compared with other cold, deep waters that still host them in smaller numbers. Is it the darkness, the lack of food reaching that depth, the oxygen levels near the bottom, or some combination no one has isolated yet? About 3,800 meters down in the North Atlantic, the wreck of the Titanic is slowly disappearing, and the cause is not rust in the ordinary sense. Since the wreck was located in 1985, researchers have documented long, rippled formations hanging off the hull that look like icicles made of rust, called rusticles. Each rusticle is fragile enough to crumble at a touch. They are built by colonies of iron-eating bacteria, including one species identified from samples taken directly off the ship's hull and named after the wreck itself. These organisms are steadily converting the ship's steel into soft, crumbling material that drifts away in the current, and entire sections of the structure, including parts of the officers' quarters, have already collapsed since the wreck was first filmed. What scientists have not agreed on is how much time is left. Some studies of the bacteria's consumption rate suggest large sections of the hull could be gone within a few decades. Other researchers, pointing to slower decay in the ship's thicker plating and its lower, more sheltered sections, argue that recognizable pieces of the wreck could still exist centuries from now. In October of 1931, the cargo steamer Beichimo became trapped in pack ice off the coast of Alaska while returning from a fur trading run. The crew camped nearby, and after a blizzard sealed the ship in ice so badly that everyone assumed it was finished, they abandoned it and made their way to safety over land. Days later, a hunter spotted the ship still floating, undamaged, sitting quietly in the ice pack as if nothing had happened. Over the following decades, the Beit-Shimo did not sink and did not stay put. Sailors, hunters, and coastal residents reported boarding her or spotting her adrift along the Alaskan and Canadian coastline again and again, sometimes years apart, always empty, always still afloat. The last confirmed sighting came in 1969, nearly 40 years after she was first abandoned, drifting in the Beaufort Sea. After that, nothing. No wreckage has ever been found, no confirmed sinking was ever recorded, and no one knows whether she finally went down in open water, was crushed and buried in shifting ice, or simply drifted somewhere too remote for anyone to notice her. In 1929, an earthquake off the coast of Newfoundland triggered a massive underwater landslide that raced down the continental slope, snapping transatlantic telegraph cables one after another as it went, and moving debris and sediment for hundreds of kilometers across the ocean floor in a matter of hours. Wrecks sitting anywhere in the path of a slide like that, or even ones caught in a slower, steady bottom current, can end up a considerable distance from where they originally went down, sometimes shifted into an entirely different underwater canyon than the one recorded in the original coordinates. This is one reason search teams looking for a specific shipwreck can spend decades combing what should be the correct stretch of ocean and still find nothing. Sediment can also bury a wreck completely within a few years, or strip it bare again just as unpredictably, depending on how the current shifts with the seasons. Oceanographers still cannot reliably predict which underwater slopes are likely to slide next, which means search teams are sometimes searching the right patch of ocean, just working from a seafloor map that describes a version of the terrain that no longer exists. According to an estimate from UNESCO, more than 3 million shipwrecks are currently resting on the floor of the world's oceans, spanning every era of seafaring from ancient trade routes to the last century. It sounds like a precise figure, but almost no one in marine archaeology treats it as one, because the number was never produced by counting wrecks directly. Only a tiny fraction of the ocean floor has ever been surveyed in any detail, and an even smaller fraction of that has been examined closely enough to confirm what is actually down there. Sonar can detect a shape on the seabed, but telling a genuine centuries-old wreck apart from a rock formation or geological debris usually needs a closer look that most sites never get. The 3 million figure is built from historical shipping records and known disaster rates, then extrapolated across regions and centuries where record keeping was patchy or missing entirely. Some researchers argue the true number is considerably higher, since so much early and non-European seafaring history left few written records at all. Others think certain regional estimates run too high. No one currently has a way to settle the question with an actual count. In 1977, a small submersible called Alvin dropped through darkness toward a volcanic ridge west of the Galapagos Islands. The scientists aboard were geologists, sent to measure heat rising from seafloor cracks called hydrothermal vents. Nobody aboard was a biologist, since nobody expected to find anything alive down there worth studying. Then the lights hit bottom, and the crew saw a scene no textbook of the time described. Dense clusters of clams, mussels, and pale worms crowded around the warm water, more life packed into a few patches of rock than most seafloor holds for miles. At that depth, sunlight never arrives, and every known food chain on the planet was thought to begin with sunlight and plants. The team radioed up for specimen jars, since they had brought none, and preserved their samples in vodka from the ship's stores. Researchers later worked out that bacteria feeding on vent chemicals were doing the job plants do on the surface, and everything else nearby was living off them. The find still leaves a question unanswered. How many more of these crowded, sunless gardens are sitting undiscovered on the dark seafloor, waiting for a light to pass over them? A living Rifsha tube worm can stretch to about 2 meters long, its red plume waving from a white tube near a hydrothermal vent. Cut one open as an adult and there is no mouth, no stomach, no gut, just one large organ packed with bacteria. The worm starts life as a swimming larva with a working gut, but once it settles near a vent and grows, that system disappears, replaced by a body built to house its bacterial partners. The bacteria take in the vent's hydrogen sulfide and turn it into usable energy, and the worm absorbs what it needs straight from them. Its blood carries a special hemoglobin able to grab both oxygen and sulfide and ferry both to the bacteria without poisoning itself, since sulfide is toxic to most animal cells. What still puzzles researchers is the handoff at the very start. Each new generation of worms is born without any bacteria and has to pick up the right strain fresh from the surrounding seawater. How a microscopic larva manages to select the correct partner out of the water's mix of microbes, generation after generation, is a step in the process nobody has fully traced. Near a hydrothermal vent lives one of the most heat-tolerant animals on the planet, the Pompeii worm, which builds papery tubes on the walls of its chimney. A single worm can sit with its tail resting in water hot enough to measure roughly 80 degrees Celsius, while its head, just a body length away at the tube's open end, sits in water closer to 20 degrees. That gradient would kill almost any other animal tissue within moments, yet the worm holds position there for its whole life. Its back is covered in a fleece of bacteria that may act as living insulation, and its blood carries proteins that seem unusually resistant to falling apart under heat. Researchers have pulled worms from their tubes to test how hot the tail end gets, and the readings keep coming back at the upper edge of what should be survivable for animal cells. How its proteins keep their shape, how its tissue avoids breaking down, and why the worm chooses to live pressed against that extreme rather than farther from the heat is not fully worked out. The Pompeii worm holds its record without fanfare, one of the plainest open questions in vent biology. A yeti crab sits near a vent crack with both claws raised, swaying them slowly back and forth through the mineral-rich water, almost like a slow-motion wave. The claws and the crab's underside are covered in fine pale hair, and that hair is really a garden, rows of bacteria growing on the strands, farmed by the crab as its main food source. Every so often the crab draws a claw to its mouth and combs the bacteria off with bristles built for the job, harvesting a crop it grows on its own body rather than hunting elsewhere. The waving motion appears to matter more than decoration. Vent fluid carries the chemicals the bacteria need, and by holding its claws in the current and moving them, the crab may be keeping fresh, chemical-rich water flowing across its bacterial farm rather than letting it stagnate. Some crabs in the same family have been seen doing something close to this same motion, even away from a vent. Whether the crabs are simply feeding, regulating the temperature of their claws, or doing something else with that steady swaying motion is still debated, and no one has settled on a full explanation for the behavior. In 2021, British glaciologists were drilling through the Filchneron ice shelf in Antarctica, lowering a hot water drill through the ice to study the ocean hidden beneath it. Their borehole was far from the open sea, about 260 kilometers from the nearest edge of water exposed to daylight, under roughly 900 meters of ice. Nobody drilling that hole expected to find much living in the water below. The assumption was a quiet, foodless stretch, too far from sunlight and open water to support life. When the team lowered a camera through the borehole, it reached a boulder on the seafloor, covered in animals, attached filter feeders that had clearly been growing there a while. There were no drifting larvae or free swimmers in the footage, just creatures fixed in place, far past where any plant or algae could grow. Everything filter feeders normally eat comes from sunlit water above, carried down by currents, yet here was a community living far past the expected limit for that delivery. What is feeding that boulder full of animals under 900 meters of ice? And how far this hidden pattern extends under the Antarctic shelves remains an open question. Drill a research borehole deep into the seafloor, past the mud and into solid rock, and instead of a lifeless core sample, laboratories keep finding living cells. This buried world is sometimes called the deep biosphere, and it stretches for kilometers beneath the ocean floor, far past any thin surface layer. These are not dormant husks either. Many of the cells tested are metabolically active, just moving and reproducing at an extremely slow pace compared to anything on the surface. Some estimates suggest that a single generation down there might take centuries, cells living in what amounts to slow motion, sipping tiny amounts of chemical energy trapped in ancient rock and sediment. When researchers add up the estimated mass of everything living underground like this, across the whole planet, the number rivals the combined mass of all plants, animals, and microbes living on the surface and in the open ocean. That comparison alone reshapes what counts as an ordinary place for life to exist. What nobody has pinned down yet is where this buried biosphere actually stops. Every deeper borehole seems to keep finding cells rather than reaching a clean bottom, and the true lower boundary of life inside the planet's crust remains unknown territory. Only 3 hydrothermal fields on the floor of the Indian Ocean are known to host the scalefoot snail, a species found nowhere else on the planet. What sets it apart is its shell and foot armor, both reinforced with iron sulfide, a mineral the snail somehow works into living tissue, giving it a dark metallic sheen unlike any ordinary sea snail. No other animal is known to build body armor this way, layering iron-based mineral into organic material grown by its own body. Researchers believe the armor likely defends the snail against crabs and other predators patrolling the vent fields, since a bite that would crack an ordinary shell struggles to get through metal-reinforced plating. But basic questions about the snail remain surprisingly open. Scientists are still not sure how it manages the chemistry of building iron sulfide into a stable shell, without the mineral flaking apart, and why a species capable of this is restricted to just those 3 fields and nowhere else studied so far. Because those same 3 fields are targeted for deep-sea mineral mining, the snail is now considered at real risk of disappearing before either question gets answered. Pull a Rimicaris shrimp up from a hydrothermal vent, and you notice it has no proper eyes on its head, none of the usual stalks or lenses most shrimp rely on. Instead, running along its back, there is a bare patch of tissue packed with light-sensitive pigment, positioned to face upward as the shrimp swarms in dense clouds around the vent openings. Vent water glows very faintly as it rises, an extremely dim thermal glow far too weak for human eye to notice, and researchers strongly suspect that back-mounted patch is picking up that light, letting the shrimp sense the position of the hottest water without needing to touch it. Thousands of these shrimp can crowd a single vent chimney, and this faint light sense may be how they keep from swarming directly into water hot enough to cook them. Proving what the patch actually detects has turned out to be difficult. The glow is so faint that separating a true thermal signal from other possible cues in that chemical, mineral-rich water is hard to test cleanly. And researchers studying the shrimp have not fully agreed on what that strange eye on its back is seeing, or how well it works. A single hydrothermal vent field does not last forever. Most run for decades before the plumbing shifts, the flow of hot mineral-rich water fades, and the crowded community around it, the worms, the crabs, the mussels, slowly starves and empties out. Yet new vent fields, sometimes appearing hundreds of kilometers away along the same ridge system, tend to get colonized surprisingly fast, arriving with worms and mussels within just a few years of forming. The animals that make this possible start life as tiny drifting larvae, released into open water with no map and no current guaranteed to carry them anywhere useful. Somehow, across stretches of open ocean where there is no landmark, no light, and no obvious trail of chemicals strong enough to follow the whole distance, enough of those larvae arrive at the next living field to restart the community there. Researchers have tracked deep currents, tested how long different larvae can survive adrift, and modeled possible chemical trails, but no explanation fully accounts for how reliably this resettlement happens. How a larva smaller than a grain of rice finds one specific patch of active seafloor among an ocean of empty rock remains one of the least understood parts of vent biology. Measure the chemical energy flowing out of many hydrothermal vents, then measure how much animal life is crowded around them, and in a number of surveyed fields, the two figures do not match. The amount of hydrogen sulfide and other usable chemicals rising from the rock, the fuel that the bacteria at the base of the food web depend on, seems too small on paper to support the sheer mass of worms, crabs, mussels, and shrimp living on top of it. By the usual math of energy moving up a food chain, some of these sites appear to be hosting noticeably more life than their measured chemistry should allow. Researchers have proposed a handful of explanations. Chemical output that varies more than any single survey can catch, extra nutrients arriving from passing ocean currents, or feeding pathways among the vent animals that have not been mapped yet. None of these has been confirmed as the full answer, and in some studied fields, the mismatch between measured energy and observed life remains sitting in the data. For now, the extra energy keeping these crowded communities fed is being treated as unaccounted for, a quiet gap in the books that deep-sea researchers have not closed. Southwest of Guam, the seafloor drops into a scar in the ocean crust called the Challenger Deep, generally listed as about 11,000 meters down, the lowest point scientists have found on the planet. Ships have measured it for more than a century using sound instead of a line and weight, since no cable could survive that trip down and back. A survey sends a pulse of sound toward the bottom, times its return, and calculates distance from the speed of sound in seawater. That speed shifts with temperature, salinity, and pressure, and at this depth the three stack together in ways hard to pin down. One expedition reports a depth, and the next, with better instruments years later, reports a figure tens of meters different, sometimes deeper, sometimes shallower. Oceanographers who do this for a living say the trench itself will not hold still long enough for a final answer. Sediment shifts, the seafloor flexes, and the tool used to measure it is being asked for precision sound was never built to deliver. Nobody working in the field today will give you one exact number for the deepest place on Earth, only a range that has never fully closed. In 1960, two men folded themselves into a steel sphere bolted beneath a vessel called the bathyscaphe tryst and let it sink toward the floor of the Challenger Deep. The descent took hours in the dark, and near the bottom one of the outer windows cracked under the pressure, a jolt the pilots felt through the hull. They reached the seafloor anyway, the first people to arrive at the lowest point of the ocean, and through the viewport, one of them later described a flat, pale creature resting on the sediment, something he took for a fish. No photograph exists, only his written account, recorded after the dive. Biologists who study that depth have debated the sighting for decades. Fish built to survive that kind of pressure had never been confirmed so deep, and some researchers now think the men saw a sea cucumber instead, easy to mistake for something else through scratched glass in a beam of artificial light. Others still defend the original account. Without a specimen or a clear image, the argument cannot be settled either way, and one of the most famous sightings in the history of deep-sea exploration remains a single unverified memory from the bottom of the world. Drop a baited camera into a trench and down to a certain depth, small pink snailfish glide into the light and pick at the bait, moving with the same unhurried ease they show in shallow water. Keep lowering the camera past about 8,000 meters, and the snailfish stop appearing, as if a wall existed that no fish would cross. Researchers have caught specimens right up to that line, never below it, and the explanation favored is chemistry rather than appetite. Snailfish cells load up on a compound called TMAO, which cushions proteins, keeping them folded correctly as pressure tries to bend them out of shape. Deeper water would need more of the compound, and at some point the concentration required would draw so much water into the cells that the fish could no longer keep its tissue in balance. That point lines up closely with where the sightings stop. Past it, the theory holds that proteins cannot be protected anymore and stop folding the way life needs them to. Nobody has caught a fish living beyond that line to test the idea, so the boundary is inferred rather than observed. From chemistry, no one has watched a fish hit. By the time an unmanned Japanese probe swept its camera across the floor of the Challenger Deep in the late 1990s, only 2 humans had ever seen that seafloor back in 1960. The footage showed pale sediment, a slow-moving animal, and something that did not belong. A thin plastic bag, the kind used to carry groceries, lying flat on the bottom. Nobody returned in a crewed vehicle for more than a decade, so for years the only human trace at the deepest point of the ocean was that bag, resting there long before a third person ever climbed into a submersible himself. Researchers only connected the dots later, reviewing decades of submersible and rover footage from trenches worldwide to catalog visible litter. Plastic turned out to be the most common man-made object at extreme depth, and that bag remains the deepest confirmed piece of it. What is not settled is how it traveled down so early, or how ordinary such a journey is. Trench currents and falling sediment are assumed to carry debris from the surface, but nobody has mapped that path closely enough to say how fast or how much litter rests in trenches no camera has reached. Lower a baited trap into a shallow bay, and the amphipods that swarm it will be small, pale, shrimp-like scavengers barely the length of a fingernail. Lower the same trap into a trench several thousand meters down, and it can come back crawling with relatives of those animals grown to the length of a human hand. The condition is called deep-sea gigantism, and trench amphipods are among its clearest examples, growing tens of times larger than shallow water cousins that are, genetically, quite close to them. It is not simply old age, since growth rates suggest these animals reach their size fairly quickly. Nor is it abundant food, since trenches are not especially rich the way a coral reef is. The leading ideas involve cold slowing metabolism, and crushing pressure changing how cell membranes and enzymes behave, either of which might let an animal keep growing past the point its relatives stop. But no one has combined those pieces into an explanation that predicts the size these animals reach. Scientists can describe the pattern precisely, list every trench where it shows up, and still not tell you why cold and pressure push a scavenger so far past its expected size. In 2022, a research vessel sat above the South Sandwich Trench, off the tip of South America, lowering sonar and a crude submersible towards seafloor rarely mapped in detail. What the crew found lay past 7,000 meters down, a new deepest point for the southern hemisphere, replacing a measurement that had stood only a few years and had itself been treated as final. The Mariana Trench gets most attention among ocean trenches, but it is not the only deep one worth naming. Close by in the western Pacific lie the Serena Deep and the Horizon Deep, separate scars cut by the same collision of tectonic plates, each carrying its own disputed numbers. What the South Sandwich discovery makes clear is how recently, and how loosely, these records have been pinned down. It is strange that the lowest point of an entire hemisphere of ocean went uncorrected until well into this century, on a planet mapped from orbit for decades. The remoteness of the trench, far from shipping lanes, explains part of the delay. Nobody can say with confidence that even this newest measurement is final, since so few sonar passes have covered that seafloor closely enough to be sure. Photograph the seafloor of a trench and scattered across the mud, you may find pale objects the size of a fist, shaped like honeycombs or fans, that look like something built by a small animal with legs and a mouth. They are not. Each one, called a xenophyophore, is a single cell, among the largest single cells known on the planet, and it builds that shell from sediment grains, sand, and tiny mineral particles cemented together from its surroundings. A single ordinary cell, the kind of unit that in most animals is microscopic and needs a lens to see, is somehow coordinating the collection and placement of thousands of particles into a structure with a consistent shape for its species. There is no nervous system directing the work, no muscle doing the placing, just one cell managing a construction far larger than itself. Biologists sort the finished shells into species by shape alone, and know the cell extends parts of itself to gather material. What nobody has explained is the coordination itself. How a single cell, without anything resembling the machinery multicellular animals use to build things, manages to produce a shape this consistent, trench after trench, specimen after specimen. Stand at the bottom of a trench, if a person somehow could, and every square inch of your body would carry a load close to 8 tons, roughly the equivalent of balancing a small car on a single fingertip. Pressure at that depth reaches somewhere around 1,100 times what a person feels at sea level, building steadily with depth. Submersibles built to survive that load are shaped like thick steel or titanium spheres for a reason. A sphere spreads crushing force evenly across its surface, with no flat panel or corner for pressure to concentrate against. Animals living at that depth do not fight the pressure with armor. Instead, their bodies hold almost no empty space inside, no air-filled cavity like a human lung or swim bladder that pressure could crush flat. Every gap is filled with fluid instead of gas, since fluid barely compresses no matter how hard it is squeezed, while trapped air would collapse instantly. That solves survival, but not everything about it is understood. Researchers still cannot fully explain how the proteins and cell membranes of these animals keep working under a load that would shut down equivalent tissue from a shallow water species within moments. Drop a fleck of dead algae or a grain of mineral dust into open ocean far from any trench, and it drifts for weeks, scattering thinly across the seafloor before it settles. Let the same debris fall into a trench, and its path changes. Trench walls slope steeply toward a narrow floor thousands of meters below the surrounding seabed, so debris landing partway down those slopes rarely stays put. It slides and tumbles further, the way loose snow slides down a mountainside, collecting on the flat bottom far below instead of spreading out. That makes trenches unusually efficient at gathering and burying carbon. Sediment cores from trench floors show higher concentrations of organic material than cores from equally deep open seafloor nearby, confirming the funnel effect is real, not just a theory about slopes and gravity. What remains uncertain is the scale of it. Trenches cover a small fraction of the ocean floor, but are so costly to sample that only a handful have been cored. Every figure for how much carbon is locked away in trenches worldwide comes from extrapolating those few samples, so the true number could be far higher or lower than current estimates suggest. When researchers wanted to test how quickly material from the surface reaches the bottom of a trench, they used a tracer humanity had already released decades earlier. Nuclear weapons tests through the 1950s scattered a radioactive form of carbon into the atmosphere, which rained into the ocean and was taken up by algae at the surface, entering the food chain from the top. When scientists later analyzed tissue from amphipods collected several kilometers down, they found that same nuclear signature inside the animal's muscle. Carbon, once part of surface plankton, was showing up inside the amphipods' bodies on a timescale of years, rather than the centuries some models had assumed material would need to reach deep trench food webs. The finding forced a rethink of how tightly connected the surface and the deepest ocean are, since a bomb test thousands of kilometers away in half a century in the past had left a measurable mark on animals assumed to live in near-total isolation from surface events. Exactly which pathway carries material down that fast, whether falling carcasses, sinking particles, or something else, is still argued over, since several routes could produce the same signature. In November 2018, seismic stations on every continent picked up the same strange signal. A steady, monochrome hum lasting about 20 minutes, too low for any human ear to register, and so evenly spread it seemed to circle the entire planet. Seismologists had never logged anything like it, and for months nobody could agree on a source, since it carried none of the sharp jolts of a normal earthquake. The trail led to the waters off Mayotte, a small island between Madagascar and the African coast, where a swarm of small earthquakes had been rattling the seabed for weeks. When the research ship Marion Dufresne surveyed the seafloor nearby, it found the answer in plain sight. A volcano that had not existed before, already 800 meters tall, built from magma drained out from deep beneath the island. The hum, researchers now think, came from that magma surging through a long, resonating crack in the crust, the way air moves through an organ pipe. What triggered the sudden draining, and why the resonance held such a pure, steady tone across such a vast distance, is still being pieced together rather than fully explained. Place a sensitive instrument in the quietest spot on Earth, far from roads or coastlines, and it will still pick up motion. A faint, ceaseless vibration with no earthquake behind it anywhere on the planet. Seismologists call this the hum, a vibration at a pitch and volume far below anything a human ear could register. Quiet traces of it turned up in recordings for years before anyone trusted the readings, and the claim that the whole planet simply rings all the time was confirmed only in the late 1990s. The leading suspect is the ocean. Waves rolling across open water and breaking against continental shelves are thought to press on the seafloor with enough shifting force to set the entire crust vibrating at once, the way tapping a drum moves its whole skin. Some researchers argue storms far out at sea matter more than coastal waves. Others suspect the atmosphere adds a note of its own. No single model accounts for the full pattern recorded so far, so the exact mixture of forces behind the steady, planet-wide tone is still argued over in papers rather than settled by them. A small earthquake breaks open on the ocean floor, far from any island, too weak to register on a seismometer standing on nearby land. And yet, on the far side of an entire ocean basin, a listening station built to detect banned nuclear explosions picks it up clearly, loud and unmistakable. The explanation lies in what happens to the shaking once it reaches open water. Seismic energy from the rupture converts into sound, and that sound, trapped in the right layer of the ocean, can cross thousands of kilometers with barely any loss of strength, arriving as what specialists call a T-wave. Hydroacoustic networks built for exactly this kind of signal now log far more tiny, ordinary earthquakes than any land-based network ever could, simply because water carries sound so efficiently compared to solid rock. What still puzzles researchers is why some earthquakes convert so much energy into this traveling sound, while others, of similar size and depth, produce barely a whisper by comparison. The exact geometry of seafloor slope and rock that turns a shudder into a clean, far-traveling tone has never been fully mapped, so plenty of these signals still arrive at listening stations completely unmatched to any confirmed earthquake at all. Lower a hydrophone into the ocean and at a certain depth, something strange happens to sound. It stops spreading outward and upward and instead gets trapped, bending back on itself again and again as it travels sideways for enormous distances. This layer, usually sitting around a kilometer down, exists because temperature and pressure change with depth in a way that creates a narrow band where sound travels at its slowest, and everything above and below bends back toward it. In the 1940s, an American oceanographer and a Soviet physicist, working on opposite sides of a war and then a long standoff, each worked out independently that this channel could carry a single sound for thousands of kilometers with almost no loss. The discovery quietly became one of the great secrets of naval science. Seafloor hydrophone networks used this same channel to listen for the faint engine noise of submarines crossing entire ocean basins. The channel still works the same way today, but its depth depends on ocean temperature. And as the oceans keep warming unevenly at different latitudes, nobody is entirely certain how much this quiet, sound-bending layer is shifting, or what that shift will eventually mean for everything that depends on it. Buried in the seafloor mud, running for thousands of kilometers between continents, are glass fibers originally laid to carry phone calls and internet traffic, nothing more. Scientists recently discovered that the same fibers, without any modification, can double as an enormous chain of motion sensors. A pulse of laser light sent down the cable shifts its properties by a tiny, measurable amount whenever the glass is stretched or jostled, and reading those shifts turns an ordinary telecom cable into what amounts to thousands of listening points strung along its length. Researchers tested the idea on a working line stretching from Los Angeles to Valparaiso, Chile, and found they could pick out the shake of a distant earthquake and the slow rhythm of ocean swell passing overhead, all from a cable never built for science. Since telecommunications companies already operate a global web of these lines, the technique could eventually turn existing infrastructure into a planet-wide sensor network without a single new instrument reaching the seafloor. What is still being worked out is how to reliably tell one kind of disturbance from another using light alone, since earthquakes, passing ships, and storms all leave signatures that, on first glance, can look remarkably alike. Somewhere along a stretch of cable lying across the seafloor, the signal suddenly stops, and hundreds of kilometers of fiber go dark at once. Repair ships are dispatched with grappling hooks to haul the broken ends up from the seabed, sometimes from a depth of several kilometers, and once the cable is examined, engineers can often read the cause in the damage itself. A clean, frayed break near a coastline usually points to a ship's anchor or a fishing trawl dragged across the line, which together account for most cable faults worldwide. Farther out in deep water, the culprit is sometimes a submarine landslide. In 1929, an earthquake off the Grand Banks triggered a wall of mud and sediment that raced down the slope and snapped one transatlantic cable after another, in order, letting engineers later calculate how fast that underwater flow had been moving. But not every break fits either story. Cables have failed in places with no shipping traffic, no known fault line, and no landslide debris anywhere nearby, leaving the recovered ends with no obvious explanation. Those cases stay in repair logs as exactly that, unexplained, long after the line has been spliced and put back into service. Off the coast of Oregon, a length of ordinary fiber optic cable laid across the seafloor has started doing something it was never built to do, recording the ground beneath it pulling slowly apart. The seabed there sits on a mid-ocean ridge, where two tectonic plates drift away from each other at a pace usually measured in centimeters a year, movement so slow it used to take repeated ship surveys, spaced years apart, just to confirm it. By watching tiny shifts in how light travels down the buried fiber, researchers can now watch that same drift happen continuously, catching a fraction of a millimeter of stretch as it happens, rather than reconstructing it afterward from scattered measurements. 20 years ago, geophysicists doubted this kind of real-time reading was achievable at all, given how faint the signal is against tidal noise. The method is now being tested at other ridges and fault zones, in hopes that watching strain build in real time might help forecast the larger ruptures that follow it. Whether steady creep like this can give warning before a major earthquake, rather than simply describing one after the fact, is still an open question researchers are working to answer. At a volcano called Axial Seamount off the coast of Oregon, seafloor instruments once recorded the ground swelling for years, then shaking in a long tremor as fresh magma forced through the crust below, with nobody there to see it. That is the ordinary condition for volcanic eruptions. By most estimates, around 80% of all magma erupted on Earth each year comes out underwater, mostly along the mid-ocean ridge system that circles the globe, invisible from the surface and mostly unmonitored. Scientists recognize an eruption underway not by sight, but by a pattern on seismographs called harmonic tremor, a long shaking produced by magma and gas forcing through narrow cracks in the rock. Axial itself is a rare exception, wired with instruments that let researchers watch its slow inflation and successfully forecast when it would erupt. Almost nowhere else on the ridge system carries instruments of any kind, so an eruption there could start and finish without a single measurement taken. Nobody actually knows how many underwater eruptions happen in an average year, because so much of the seafloor is never being watched at all. Drag a magnetometer behind a survey ship across the mid-Atlantic, and the readings jump back and forth in a regular rhythm. Stripes of rock magnetized one way lying beside stripes magnetized the opposite way, like a barcode printed across the ocean floor. The stripes form because new crust is constantly created at mid-ocean ridges, and as the molten rock cools, its magnetic minerals lock into whatever direction Earth's field happened to be pointing at that moment. Since the field has flipped, north becoming south and back again, many times over the planet's history, the seafloor records every flip in order, moving outward from the ridge like rings on a tree. Two British geophysicists published this explanation in the 1960s, though a Canadian scientist reached the same conclusion around the same time and saw his paper rejected by two journals before the idea was credited to others. The stripes are now considered settled science, one of the strongest proofs that the seafloor spreads at all. What nobody has explained is why the field reverses at all, or why the gap between one flip and the next can run from tens of thousands of years to tens of millions, with no obvious pattern. Pull up a modern satellite map of Mars, and it will show mountains, craters, and canyons in far sharper detail than most maps of the ocean floor here on Earth. Only about a quarter of the seabed beneath our own oceans has been surveyed directly, ship by ship, using sonar beams that bounce off the bottom and build a true picture of what is actually down there. Everywhere else, the maps people rely on are filled in by estimate, built from satellites hundreds of kilometers overhead that measure faint bumps in the sea surface, caused by the gravity of whatever mass sits on the seafloor below. It is a workable stand-in, and it gives a rough shape to the planet's seabed, but it cannot show anything small. A shipwreck, a single seamount, a narrow canyon, anything under roughly a kilometer across disappears from the picture entirely. An international effort now aims to survey the whole seafloor directly before the decade is out, gathering data from research vessels, volunteer ships, and autonomous drones sent to fill the gaps. Until that project finishes, nobody can say exactly how many mountains, trenches, or other features on our own planet remain completely uncharted. Off the eastern coast of Australia, researchers spent years recording the same humpback whale population every breeding season and kept finding the same pattern. Within any one season, every male singing in those waters used almost the same song, phrase for phrase, as though the whole population were performing from a single shared score. A season or two later, that version was gone completely, replaced by a different one, usually a song first recorded off the western side of the continent in the Indian Ocean. Tracking it over several years, researchers watched a new song type appear in the west, turn up in the east the following year, then keep traveling, reaching whales singing near French Polynesia a year or so after that, far out across the Pacific. Nobody has found a courier for this. No group of whales swims the whole route carrying the new version along from population to population. Populations thousands of kilometers apart simply end up singing the same tune within a couple of years, and the direction never changes, west toward east, never the reverse. What sets that direction, and how animals scattered across an entire ocean settle on one arrangement so quickly, has no accepted answer yet. Only male humpback whales sing. That part biologists settled decades ago, listening to recordings made on breeding grounds in winter, where singers hang alone in open water, head down, repeating a set arrangement of moans, cries, and rising phrases that can run for close to an hour before starting over from the beginning. The obvious guess was that singing helps attract a mate, the humpback equivalent of a bird's dawn chorus. That explanation runs into trouble fast. Females have rarely been seen approaching a singer, let alone choosing one singer over another. Other males, though, often swim straight toward a singing whale, suggesting the song may be doing something between rivals rather than performing for an audience of females at all. Neither idea explains why the song needs to be so long and elaborate, or why it keeps changing season after season, when a much simpler, stable signal would announce a male's presence just as well. Researchers can describe exactly how the song is built, note for note, and still cannot say with any confidence what problem, in evolutionary terms, all that length and constant revision is actually solving. Sperm whales do not sing the way humpbacks do. They communicate in short bursts of clicks, called codas, fired off in patterns that sound almost like a code passed between animals resting at the surface. Listening across different populations, researchers found that sperm whales sort into clans, each with its own set of preferred coda patterns, distinct enough that a trained ear or a computer can tell which clan is speaking before knowing anything else about the whale. What surprised researchers studying family groups near the Caribbean islands was how a calf comes to use one pattern over another. It does not simply copy its mother's rhythm. Calves raised within a clan pick up the clan's coda style even when their mother leans slightly differently, as though the group, not the parent alone, sets the dialect a young whale grows into. Clans with different dialects often share the same stretch of ocean, feeding in overlapping waters without blending their patterns, generation after generation. Why the boundary holds, what keeps neighboring clans from mixing their codas, the way related groups of other species often do, remains an open question in a field still learning how to listen. For most of the time people have studied sperm whales, nobody knew how, or even whether they properly slept. The animal dives to enormous depths hunting squid and needs to keep surfacing to breathe, which made deep, sustained sleep seem almost impossible to fit in. The answer came by accident. A research vessel moving quietly through open water once drifted straight into a group of sperm whales that never reacted, never moved aside, floating upright in a loose cluster, heads near the surface, bodies hanging straight down into the dark like a set of pillars. Only when the hull actually touched one did the group startle and dive. Tags later attached to sperm whales confirmed what that encounter suggested. The whales enter short, motionless drifting bouts, upright in the water column, taking up only a small fraction of their day, far less sleep than most mammals need. It is apparently enough for a brain and body that size, but nobody studying the tag data can say exactly how such brief vertical naps deliver whatever rest a sperm whale requires, or why the animal chooses that upright posture to do it in. Every population of blue whales monitored anywhere in the world's oceans has been singing lower, year after year, for as long as anyone has kept recordings. Listening stations tracking the deep, drawn-out calls blue whales use over long distances have logged the same slow downward drift in the Pacific, the Atlantic, and the Indian Ocean, populations that never come into contact with each other, all sliding toward lower pitch on roughly the same schedule. The shift is small from one year to the next, barely noticeable in a single recording, but laid end to end across the archive, it adds up to a steady decline with no sign of leveling off. Researchers have tested the obvious explanations. A recovering population, further from the brink of extinction than it was under commercial whaling, might change how males compete for attention, but that does not obviously predict a lower pitch. Rising background noise from shipping might push whales to alter their calls, but the timing and worldwide consistency of the drift do not line up cleanly with noise trends. The decline keeps being documented far more precisely than it is being explained, a strange position for a measurement this well established to sit in. During the Second World War, sonar operators kept detecting a false ocean floor, a solid-looking layer that reflected sound as if the seabed had risen hundreds of meters toward the surface. By day, that layer sat deep and stayed put. By night, it climbed. It took years to work out what the layer actually was, not rock or sediment, but an immense, loosely organized crowd of small fish, shrimp-like creatures, and squid, rising together as the sun went down and sinking back before it returned. This nightly climb toward the surface is now generally described as the largest synchronized movement of animal life on the planet, dwarfing any migration on land, repeated every evening rather than once a year. Whatever boat sits on the open ocean after dark is very likely floating directly above the most crowded layer of animal life on Earth at that moment, with no visible sign of it from the deck. The leading explanation is that the climb trades safety for food, feeding under cover of darkness rather than in daylight where predators see clearly. Researchers still cannot fully explain why different layers time their rise so differently, or what exact combination of cues sets the moment each evening. Somewhere near the bottom of the northern Pacific sits water that has not touched open air since roughly 1,000 years ago. It sank in the far North Atlantic, part of a slow global circulation that pulls water into the deep ocean and drags it, at a walking pace measured in centimeters per day, across the planet along the sea floor. By the time that water reaches the North Pacific and rises again, whatever was happening at the surface when it sank has passed out of living memory everywhere else. The water carries a kind of record with it, gases, minerals, and dissolved matter absorbed from the atmosphere, and the surface ocean at the moment it went under, sealed away from air and light the whole way. When it eventually upwells, it releases what it picked up 1,000 years earlier back into contact with the atmosphere and the creatures living near the surface today. Oceanographers can date this water fairly precisely using the chemical traces locked inside it. What is harder to pin down is the full account of everything that water absorbed on its way down, centuries ago, and how much of that old, sealed-away chemistry still shapes the water rising today. Below the reach of sunlight, roughly a kilometer down and continuing far deeper still, lies a layer of ocean known as the midnight zone, permanently dark, cold, and enormous. Weigh its volume against every other habitat on the planet combined, forest, grassland, coral reef, and shallower ocean layer alike, and the midnight zone still takes up more space than all of them. It forms, by a wide margin, the largest living space on Earth, simply because so much of the planet is ocean, and so much of that ocean is deep. And yet almost none of it has been directly seen. Cameras and submersibles have covered a smaller fraction of the deep ocean floor than spacecraft have photographed of the surface of the moon, a comparison that keeps holding up no matter how many new expeditions launch, because the ocean simply keeps being that large. Every dive into that layer runs on limited time, a few hours at most before the vessel has to surface, inside a volume that would take, by some estimates, centuries of steady exploration to properly cover. Nobody studying the midnight zone claims to know what fraction of it has truly been looked at, only that the number is small. Across the history of ocean science, from the first dredges lowered over the side of sailing ships to the newest remotely operated vehicles, the total area of seafloor actually sampled remains a small patch by any measure, scattered thinly across a planet that is mostly ocean. Whole stretches of seabed the size of countries have never had a single scoop of sediment brought up, let alone a camera pass overhead. That patchiness shows up constantly in practice. Research teams sending a submersible to a seamount or trench that has never been sampled before routinely come back with something that matches no existing description. A worm, a shrimp, a fish built for pressure and darkness no field guide anticipated. It has become an expectation rather than a surprise among deep-sea biologists that a first dive to a new site will turn up at least one organism new to science. Museum collections are still absorbing the backlog, specimens sometimes waiting years for the right expertise to formally describe them. Nobody working in the field will estimate how many more such organisms remain to be found, only that the fraction of seafloor examined so far gives no reason to think the list is near finished. Every night in the open ocean, a vast crowd of small fish, shrimp-like animals and squid rises out of the deep and drifts up toward the surface. And every night, before the sky shows any real light, that same crowd sinks back down again, unhurried, finishing well before sunrise makes the surface too bright for it. By the time the sun is properly up, the water near the top looks the way it always looks in daylight, calm, clear, apparently empty. Nothing in view to suggest that anything had gathered there only hours before. That emptiness is the most misleading thing about the open ocean. For as long as people have sailed across it and looked at flat water and seen nothing, the assumption seemed reasonable. Not much lives out here, away from reefs and coastlines. Only continuous listening and tracking revealed this nightly gathering inside that apparent emptiness. And even now, after decades of surveys, Nobody has settled on a firm total for how much life makes that daily round trip. Estimates of the total weight involved have shifted by large multiples as measuring methods improve, and the true scale of what passes beneath a quiet hull each night is still, technically, unmeasured. That is 100, and the water has gone still. Every one of them is still open. Somewhere tonight a ship is drifting with its engines off and a cable running down into the dark, and someone is watching a screen, waiting, the way people have waited for a very long time. None of it needed solving tonight. It was only ever there to be wondered at. The last of the layer is sinking back down now. The surface above it is flat and empty and gives nothing away, the way it always has. If this kept you company, there is a new one of these every week, and it will be here whenever you need it. Sleep well. The ocean will still be there in the morning, and so will everything we have not worked out about it yet.